Hello viewers, now I am going to talk today about cell signaling. As we all know, cells are the basic structural and functional unit of all the living beings, and the basic qualities of living beings are to reproduce, that means to produce their own kind, and to respond accordingly. Understood? So since there is a all the living beings responds accordingly to their immediate environment or external environment, therefore they must be able to understand or receive some signals and then accordingly respond. So the significance of signaling, or to receive signal and to give response, is of utmost importance in all the living beings present in this world. Now, unicellular individuals and multicellular individuals, both of these kind of individuals, they exhibit this common phenomena of cell signaling or exhibiting response, because both this kind of individuals respond accordingly. However, the response of an unicellular individual is basically, since they are made up of single cells, so they respond by giving just in a kind of response of a single cell manner, and in case of multicellular individuals, the response are in a lot of integrated sense. That means the response is in sort of an integrated response. So just let us think about an amoeba, how an amoeba responds to a signal. So a very basic question may arise that amoebas, they are protists, so do they have sense, or do they exhibit consciousness? The answer is yes, although they are individuals, unicellular individuals, since, but they also have consciousness. That means they have a sense of sensing the signals and accordingly giving the response. That is, if you give an chemoattractant molecule to an amoeba, it will go towards that chemoattractant molecule. That means it will exhibit chemotaxis. But if you give a chemorepellent molecule, you will distinctly observe that the amoeba goes simply the opposite way of that chemorepellent. Understood? Now, in case of multicellular individuals, the response is also very evident. Now, let us take the example of adrenaline, the hormone. As we all know, the hormone responsible for giving fight and flight response. So these kind of hormones, they are basically released due to stress. Now suppose you are walking nearby and suddenly you see a tiger. So what would happen? So that would evidently act as a stress to your neurons or your nervous system. That means you will perceive that signal as stress, and accordingly that adrenaline which would be released due to stress, that I am not going into the details. So they will be released into the bloodstream, and they will go to different kinds of effector organs, giving differential responses to all the cells of these different organs. And ultimately what will occur, as suppose you have seen a tiger, so what would happen? Suddenly you will notice your heartbeat will increase, and suddenly you have started to take up upon very deep breaths and very frequent breaths. So this is what occurs, and why this occurs? Due to give high amount of nutrition and high amount of oxygen to your cells, mainly your muscle cells, in order to simply run away from that situation. Understood? This is why your heart rate increases, and to give rise to heightened pressure, so that all the cells of your deep cells and all the muscle cells get high amount of oxygen, and in order to get oxygen you need to have heightened amount of respiration. That's why the bronchioles dilate, and the liver breaks the stored glycogen into glucose to give you a lot of amount of energy, and this is why the different cells give a kind of integrated response so that you can fly. So this is the significance of cell signaling. Also, the multicellular individuals, since we exhibit division of labor, so the different organs and different cells have become specialized to perform a simple or particular task in an integrated manner to give a response as an individual. So in this manner, in order to respond accordingly to any kind of situation or stress or signal, the individual have to act as a one, and all the different cells or all the different tissues and organs and systems has to act accordingly in an integrated manner. So here comes the question, who is that integrator? Now the answer is very simple. The integrators are the signals. They are released from different kinds of cells in order to integrate this kind of all the organs. A very well known example is endocrine system, which is a chemical integrator. Another so these kind of signals or hormones, they integrate our body and to give response as a whole. There are different kinds of other signals that integrate us. I am not going into the details. Now what are the different broad types of signaling? So as far as any basic different kinds of signaling is concerned, there are different kinds of signaling. One of them is juxtacrine signaling, or you can say contact dependent signaling, where the signaling cells or the sender gives a signal and there is a target cell which is known as the receiver, who receives the signal by means of a receptor or receiver. Understood? So, and these kinds of juxtacrine signaling is very evident during immune responses and also during developmental processes. There is another kind of signaling which is paracrine signaling, where the sender gives the signal which is diffusible and which diffuse from one region to another, and this diffusion is very local. That means in a very adjacent situation or adjacent region where the receiver cells receives the signal and accordingly gives response. There is another kind of signaling which is very known to which is very well known to all, that is endocrine signaling, where the endocrine cells secrete their product or hormones into bloods, and the signal or the hormones, they flow from one region to another region through blood and goes to the target cell and gives and binds to its receptor or receiver and accordingly gives a response, which is a very well known fact, as I have given you the example regarding the adrenaline fight and flight example. Another very important aspect is the synaptic signaling, which is a very specialized kind of signaling, which is neither contact dependent nor endocrine nor paracrine, where the cell or the presynaptic or the synthesizing cell or the sender cells have long processes, and it releases the signal very close to the target cell. The target cell or the next cell, it may be a neuron or it may be a muscle or it may be a gland, whatever it may be. Now the receiver receives the signal and it elicits a response. Understood? So these are the basic broad types of signaling. Now why paracrines are in local or local mediators, or why synaptic, what are the difference between synaptic and endocrine signaling, I am not going into the details. If you wish, you can always ping me. I can provide you those basic principles of why paracrine is localized and endocrine and synaptic, what the difference between synaptic and endocrine signaling, and that's all. Now what are the basic principles? Now in order to respond accordingly, since we are very what we should say that we are very advanced forms, so we are advanced form because we exhibit very much of complexity. complexity regarding organizations, complexity regarding body structures, and complexity regarding responses. Now, to give a complex response, there is a basic mechanism, that means to screen the environment according to different messages or signals and accordingly give rise to a particular response. Now, in a normal situation, a particular cell, let us take let us for example take the cell which is present in our arm and which is present in deep tissue. So, though that cell, maybe that cell at a particular time receives a different kinds of signal, because it receives the blood-borne tissue factors or the blood-borne different types of growth factors and other kinds of molecules always always continuously in a particular time span. So, since it receives blood every time, that means blood has a mixture or heterogeneous mixture of different kinds of molecules, that means it has different kinds of signals. Now, if it receives a specific kind of stimuli, that means suppose A, B, C, different kinds of factors, it would exhibit survivability, because that message or that encoded sets of message would give rise to or ultimately binds to the receiver, and the receiver or the responder or the receptor activates the cell and gives rise to different kinds of signals. Similarly, different kinds of signals which act in according order to gives rise to, always it gives rise to a specific response. So, this is the key point, that means that each and every kind of signal should have a specific meaning, therefore, it would be decoded by a specific receiver, that means the receptor, and it will give a particular kind of definitive response. There means if I go into the molecular mechanisms, so therefore, the different molecules involved in cell signaling are of different types. One of them should be one which is a very important one, that is the receiver, or which we call as the receptor. The receptor, as we all know, it receives a signal or a signaling message, this red dot, which we can say as the ligand or signal or message, whatever you may whatever you want to say. But this signaling molecule, suppose a hormone or anything else, they are basically encoded messages. These encoded messages binds to the receiver or the what we should say the receiver or the receptor, and this receptor decodes it. So, this is the decoder, and it decodes the signal, and ultimately it activates a different sets of molecules, different sets of intracellular molecules, which ultimately activates the effector molecules, or one which exhibit effect. And these effector molecules, they exhibit different kinds of functionality, and ultimately these functionalities are exhibited as response, which we have seen in these cases. Understood? Now, so how or what is the basic mechanism of signal transduction? As we now let us draw an analogy. So, as we all know, this string doll, now as we all know that this doll dances accordingly, okay, and this dancing of this doll is dependent upon how you make it dance by this pulley, and how the strings are attached with, and which strings are attached with and definite parts of this doll. So, suppose you want to make the hands, so you want to make, or you want to simply move this doll's hands. So, what we need to do? First, you need to move the specific part of this pulley, and which is attached to this hand. Am I clear to you? Now, since the particular movement of this hand is not only dependent upon how you move this pulley, but also how the strings on which strings is attached to this arm also, isn't it? So suppose you just move this pulley. Suppose you have just you try to move this pulley, but you see no movement of his hands or its legs. Why so? Because the strings may have been teared, or the strings are not maybe not attached to this particular hand, isn't it? Similarly, the receiver, which is this pulley, the receptor exhibit response by different mechanisms, not only by the receptor himself, but also the molecules which are also involved, which the receptor activates one after another, which we called as downstream molecular cascades. So a signal transduction pathway or signaling cascade, as we all know, this signaling cascade is also very important. Why? A signal exhibits a particular kind of response. Am I clear to you? And this there are different kinds of molecules. There are relaying molecules, amplifying molecules, integrative molecules, anchorage molecules, modulators, spread, scaffold proteins, and effector molecules. Different kinds of molecules, they exhibit different kinds of functionality to ultimately give a specific response. And who determines which molecule will be activated? That is the controller, or you can say the receptor, that determines which particular molecules would be affected, and after activation through signaling, the receptor relays this activation one after another to give rise to a specific response. Now, what is a receptor? As we all know, the signals are basically encoded messages, and the receptor is a decoder, and this decoding is basically done by a simple mechanism. That means a simple non-covalent interaction between the receptor and the ligand gives rise to a different kinds of chemical changes inside the cell, which we call as signal transduction, which ultimately gives response. And the specific route of this from receptor to response, that means the specific class of molecules which are activated one after another, just like this particular specific route is, or the particular way of activation is known as the pathway, or the particular signaling cascade which they follow, or which they activate one after another to give rise to or to elicit a response. So that means a receptor is you can say it's a response evoker. It evokes a response whenever the signal is binds to it. So a signal, you can simply draw an analogy by signaling what you can say an antenna which receives a signal is the receptor, and the center from which these waves are emitted, they can be simply said or simply visualized as a signaling molecule. So a sender cell, just like a sender of different kinds of radio waves, the sender cells sends this waves or signals or encoded messages, and through these antennas or receptors, the receiver cell or the target cell receives this signal and ultimately gives a response. However, those cells which binds to a signal and gives a response, they are known as the target cells. And there are lots of cells which are non-target cells. Why? There are two mechanisms or two possibilities why a cell is a non-target cell. The mechanism may be the absence of a receptor, that means the absence of an antenna which does not emit any kind of sound through the radio. So suppose a radio which is not working. What are the basic problems maybe since why the radio is not working? One, suppose the one possibility may be the receiver is not working, that means it is not receiving the signals. Another problem is, suppose the receiver is okay, but the internal mechanism of the radio is broken, isn't it? Similarly, a non-target cell may be a non-target cell because the absence of the receptor or the absence of these downstream molecules. Both of them are possible. Now, what are the properties of this signal transduction? One is specificity, that means the signaling molecules, they should be, or the ligand should be very specific to the receptor. So, since the concentration of signaling molecules, suppose let us take the endocrine pathway or endocrine signaling, the receptor, the signaling, the concentration of signaling molecules are very low. So, these very low kind or with this very low, with this very low concentration, the receiver or the receptor can evoke a very potent response. Such was in the case of adrenaline. So, the another background property of signaling is to amplify. That means a receptor has to amplify or has to activate a protein or proteins that would amplify these responses, and these kind of proteins are basically known as amplifier, okay, which amplifies the response and basically induce a heightened amount of response by just means of single molecular binding. Understood? So, this kind of amplification is basically done with by the means of second messenger systems. Now, another thing is, since there are different kinds of molecule inside a cell, okay, and the different kinds of receptors and different kinds of signals, so therefore to maintain a specific route, the specific downstream molecules, they are arranged in a specific module. That means this module assembly is basically mediated by different kinds of scaffold proteins, which ensures that different kinds of proteins are present in a clump or in a group in order to simply relay one or simply activate one after another by simply ring activation through the receptor. Another very important aspect is desensitization. That means whenever, in order to respond accordingly to different kinds of signaling, you must be able to sense the concentration of the signal or ligand very efficiently, and in order to do so, you must be able to exhibit desensitization. That means whenever a signal activates a receptor, it should incorporate a negative feedback loop to also inhibit itself in order to specifically sense the concentration of different kinds of molecules or different kinds of signals, and to simply neglect sensitization, which is basically a sense of nullness or a sense of inactivation in presence of the signal. So, they must be they should anticipate this kind of sensitization, and they add in active manner to desensitize this kind of transduction. Another very important aspect is integration. That means since different kinds of signals are activating a particular situation in a particular cell, so therefore different kinds of molecules are activated, and the different kinds of molecules activated, they must be integrated in a specific sense in order to give give kind of a or give rise to a particular definitive response. And another very important, last but not least, another very important property is localized response. That means when the signaling molecule binds to the receptor, immediately the signaling molecule is destroyed, or somehow the signaling molecules is inactivated so that it cannot activate further molecules in order to give a particular localized response. So, these are the 6 basic properties of all the basic kinds of cellular signaling mechanisms. Now, since we are talking about the receiver, which is a very important attribute of this signal transduction pathway, the receptor may be depending upon the ligand or depending upon the chemistry of the ligand, the broadly we can classify receptor upon depending upon the ligand affinity into two classes. One of them is a cell surface receptor or extracellular receptor, another one is intracellular receptor. The cell surface receptor is basically present in the surface of the cells, that means in top of the cells on the plasma membrane, and the intracellular receptors, they are present inside the cells. They may be they may be present inside the cytosol, or they may be present inside the nucleus. So there are two kinds of, basically there are two kinds of receptors depending upon ligand chemistry. Now here comes the twist, that what are the basic or what are the basis of this kind of receptors and why these two kinds of receptors are present. Now since our cell is covered by a plasma membrane which is basically hydrophobic, and the plasma membrane basically differentiates between two cellular or two aquatic component, one which is present inside the cell, the intracellular component or cytosol, and one which is present outside the cell membrane or the extracellular fluid or the interstitial fluid compartment. So in order to, so there are two such fluidic compartments or two such polar compartments which are differentiated by this plasma membrane, and suppose the signaling molecules are basically soluble in water. So there may be two kinds of molecule, one depending upon solubility, one they may be soluble in water, so they are hydrophilic, lipophobic, and two they are not soluble in water, suppose steroids. So they are not soluble in water, that means they are hydrophobic, that means lipophilic. So being hydrophilic, there is an advantage there, so they can easily solubilize in blood or whatever interstitial fluid, and they can flow from one region to another region. Then since they are hydrophilic, that means they cannot pass the plasma membrane, isn't it? So this, since the message cannot pass the plasma membrane, the receiver should be present in such a way that they can bind to the receiver and activate receiver or the receptor, so that signal transduction can efficiently occur. So due to that, the hydrophilic molecules, they bind to extracellular receptor, it's very common sense. And since the lipophilic molecules or hydrophobic molecules, they can easily cross the plasma, or they can easily diffuse away from this plasma membrane, diffuse through this plasma membrane. So therefore the receptors, they can be placed inside the cells, in cytoplasm or nucleus, whatever it may be, and in cytoplasm the signal goes to it, or the ligand binds to the receptor, it binds to the receptor and it activates it, and it elicits a particular kind of response. So because of the solubility or because of the chemistry of the ligand, there are two kinds of, broadly we can differentiate upon as extracellular receptor and intracellular receptor, or hydrophobic molecule bound receptors and hydrophilic molecule bound receptors. Now the extracellular receptors, they can be broadly classified into multiple classes. They can be classified into a G protein coupled receptor, that means a receptor which elicits response by a molecule which is known as a G protein, that means a GTP or GTP binding protein which have an inherent GTPase activity, and this after receptor binding they activate this particular class of protein which is known as G protein, and this G protein activates further class of enzymes and activators or effectors that elicits a particular response. Another kind of receptor is very important. They are receptors which are associated, they are directly associated with enzymes. Since this G protein coupled receptor exhibits their activity not directly activating enzymes, rather they activate the enzymes by means of a mediator which is a G protein or G protein or GTP binding protein. There are another kind of receptors which directly activate an enzyme. There are two kinds of things. Suppose the receptor itself is an enzyme, or it is associated directly with an enzyme. So, there are two such possibilities, and this kind of receptors, they also exhibit a very potent kind of signal transduction pathway. Another kind of receptor is present in we say that ionotropic receptors. That means the receptors which are basically themselves are ion channels, and signal after binding to the receptors activate these ion channels, and the ions move according to their electrochemical gradient. Understood? As far as the nuclear receptor or intracellular receptors, these kind of receptors, they are basically present inside the nucleus. In absence of the ligand, they act as a repressor of the particular responsive genes, and when the ligand binds to that particular receptor, they become activated and they become transcription activator, and they activate a particular kind of expression of particular target genes. So, this is how they exhibit a particular kind of genomic response, these intracellular receptors. Understood? So, so there are different kinds of ligands, as I told you earlier. Depending upon their biochemistry, they would have different kinds of receptor located in different kinds of regions of a cell. Now, a basic question may arise that why a particular kind of signal exhibit different effects in different kinds of tissues. Now, the answer is very easy. One probability, as I told you before, a cell which is responsive to a particular signal, the basic mechanisms are depending upon the receptor and the downstream molecular pathways. So, a signal which binds to a particular cell and give different kinds of responses is entirely dependent upon which receptor is present and which are the downstream molecular pathway they have undergone or activated. Since, let us take the example of acetylcholine, which ultimately decrease the rate and force of contraction in cardiomyocytes. In skeletal muscle cells, it exhibit high amount of contraction, and in case of salivary glands, it exhibits secretion. So, since different kinds of responses are activated, that means different effector molecules are activated due to this particular signal, isn't it? So, now this different kind of responses exhibited by a single signaling molecule thus ensures that the differential or tissue specific response is due to either different kinds of receptors they exhibit, that, or they may be same kind of receptors but different kinds of downstream molecular pathway. So, suppose why they have different